I was a graduate student here working with Tom Knight at the time, and I got interested in synthetic biology around 96 or so. And I spent a lot of time in the beginning, obviously trying to engineer bacteria. That was much easier to deal with, much more known about bacteria at the time. But even from day one, I knew that I wanted to be able to engineer mammalian cells. And it's just been fascinating to see the developments in synthetic biology in bacteria over the last decade or so, but over the last maybe 3-4 years to see really the explosion on what we can do in mammalian synthetic biology. So in terms of this notion that we often see in synthetic biology, the bottom-up notion of taking devices and creating modules out of them, and this really, as I said, we've seen so many applications, so many different circuits and topologies being created, and now really allowing us to think about creation of systems. And there's obviously a fuzzy, there's no strict line that delineates where modules end and systems begin. It's more of a fuzzy notion of scalability, but we are now able to explore specific applications that we believe are uniquely enabled by synthetic biology. So it's been fun to go from just being able to turn GFP on and off to actually hopefully being able to do something useful with the cells. We often see this notion of a bottom-up analogy for synthetic biology. It's not true. Synthetic biology is not really a bottom-up discipline. We do bottom-up assembly, but many of us really want to think about also the top-down aspects of synthetic biology. And so I teamed up with Jake Beal, who was here and who was also a graduate student with me back in the 90s, and also with Doug Nensmore, to create this workflow where we're trying to formalize the process of going from some kind of high-level design all the way down. So high-level ideas of what you want to be able to design, formally describe them with some perhaps a programming language. It could be eventually something else, maybe 3D CAD tools, if you will, and have a compiler that takes it all the way down to something that we can actually put into cells. So I'll give you a very simple example. So we can write code, and the reason we write code like this is Jake and I, we were in this graduate program together where we used Lisp. So there's a whole bunch of parentheses. So there's just by accident from that. But if you don't like a whole lot of parentheses in your programming languages, this is another representation. If the input is high, produce cyan fluorescent protein, else yellow. So the point is that we can formally encode that, and then the biocompiler basically goes through a series of steps. This is a data flow, and then comes up with an abstract genetic regulatory network where we have some of these elements not defined yet, and then we have another element called Matchmaker that was developed a lot by Doug Densmore, Fusen Yemen, and also Jake, to figure out what parts actually match the appropriate requirements that we have. And so this gets automatically created, and also robot instructions, so that ultimately, the graduate student, the postdoc, or the undergrad of the future won't really have to know how to do cloning, right? So that we have a robot that actually does all of this work for you. And so this is actually reality in some of the synthetic biology entities out there, some companies and some academic institutions that have some funding for this. But the hope is that ultimately, microfluidics. So everybody has a microfluidic device on their bench where they don't really have to worry about this. This becomes mundane, but understanding how to go from here to here is really where the interesting thing lies. I want to point out that we want to certainly be able to use computational design tools. And again, this is work with Fusen Yemen, Aaron, and Jake, where this notion of being able to write high-level programs, and this appears to be complicated in the beginning, but then the biocompiler can take these relatively complex programs and then automatically generate something that hopefully is easier to implement. So there's a big difference between for a graduate student, trying to implement something that has 15 regulatory units to something that actually has 5. So you may have a chance of graduating, in your lifetime and so on. And then especially as we wanted, even if we do come up with 5, which is the correct 5? So if we wanted to create 1 or we wanted to create 1,000 of them, certainly things like this become quite important. So what I want to talk about now is basically coming up, with at the foundational level, and then trying to build up ultimately to application. So with mammalian synthetic biology, we need to be able to build things, and size matters. So we want to be able to build large circuits because they allow us to encode much more sophisticated mechanisms. So here's work with Patrick, Lila, Yingqing, and Xavier to be able to very quickly be able to come up with circuits. So we adapted the Gibson assembly. So probably most of you here certainly know about Gibson, or at least may have done experiments. So we adapted that in particular to have the ability to focus on mammalian components. And so now we can take Gibson assembly and create single circuits very quickly. So within a matter of 5 days from the time we decide which parts we want to have to the time when we actually have a single plasmid that has all of these parts together arranged, hopefully in a reasonable way, is really now a process of 5 days, and it's actually become quite reliable. And we can create routinely things that are on the order of 60, we've had 61, 64 kb. So this is now appears to be not as much of a bottleneck as it used to be. Okay, so now our ability to actually design something that may work is becoming a bottleneck, and also other things like parts that work, and how do you get everything to match up together. So that's been important. The other thing, and Xavier, I believe, has a poster on this as well, is we want to be able to get the DNA in there in various ways. So we do a lot of transient transfections, but oftentimes we want to be able to have stable chromosomal integration, and we want to be able to do that efficiently. So in this particular case, Xavier developed a mechanism that uses the BXB1 recombinase, and when we use that recombinase, we have very nice efficiency. So within a matter of 72 hours, without selection, 10% of the population have a single integration of the plasmid. Okay, so that really allows us to, very quickly, maybe not quite as fast as bacteria, but not necessarily a whole lot slower, is to be able to introduce things into the chromosome. They're in there in a single copy. They will be there. They remain there probably longer than the experiment itself, and they hopefully won't get mutated as far as we know. And so the recombinase that Xavier's used, the BXB1, seems to have quite increased efficiency over existing ones. And that really allows us the increased efficiency and the ability to create large circuits, I think, are really changing the things that we are able to do in mammalian synthetic biology. So for example, we can build libraries rather easily now. So here's a library that we built of different fluorescent proteins that are targeted to different places. And we see that we have a one-pot reaction. We have a library of various genetic elements, genetic circuits. We integrate them stably into the cell lines, and we can see that all of them are in fact part of now of these polyclonal populations. So that really decreases the time. We don't have to now worry about making single clonal populations for many of the experiments that we do, and we see that we can create very large diversities of the circuits that we actually want to test, and we're now working on creating much, much larger libraries than this, like up to 10,000 components or so. We can also integrate things that are quite large now in a reliable fashion. So this is a 27 kb. This is a two-step process where we create a landing pad with zinc finger in the nucleases first, and then we use the BXB1 recombinase to put in these large circuits, and it works quite well. And also the circuits often function. Certainly not always, but they do sometimes function. So here's a fairly large circuit where we have stable single integration, where we see initially cerulean. Then we induce expression of YFP and also another recombinase, and we see that here. And as a result of that, we actually have recombination happening to the synthetic circuit that we put in there. We see that with yellow expression here. We see that the red expression goes up, and then we take dox away. And after about 14 days, a 14-day experiment, we see very nice red fluorescence, and then the yellow fluorescence goes away because there's no longer the DOX inducer. So the other thing that's nice for those of you that may not see FACS histograms quite often of fluorescence in mammalian cells, these are nice and tight. Okay, so we often get into a problem of large heterogeneity, especially with transfections. So we see two orders of magnitude variations in fluorescence, and that makes it very difficult to actually reliably engineer things. And see here with the stable single integration that the distribution are quite nice. So as an engineering nerd, I get really excited about things like that. So we can build large circuits. Gene expression works quite well, and we can now integrate quite reliably and quite quickly very, very large circuits as well. So now the question becomes, can we predict what's actually going to happen? And this is clearly an important thing in synthetic biology. So this is work with Noah, who is my lab, and now in Church Lab, and also with Jake. And so the question is, and this is something I've been trying to do for a very long time, is actually this very simple notion that people that come from the outside assume just works. And everybody that's in synthetic biology knows it doesn't work. Is can you characterize one part, characterize another part, put them together, and predict what happens? Sounds like a simple thing to do, and everybody assumes this, and then they realize we don't tell them right away that it doesn't work, right? We want to suck them in and have them be invested in the technology first. So I've been trying to do this. I've been trying to do this, for example, in bacteria with cascades, and it didn't work. Oftentimes, what we try to do is we measure the parts. We build computational models. We do parameter estimation. There's never quite enough information to really give you a perfect model, right? And then we do predictions, and they don't work. And so then we slightly revise the paper, and we say, the model was used as a guideline to understanding the circuit behavior. And I certainly have done that quite a bit myself. And so we said, can we try to do better? And so we said, it didn't work in bacteria. Let's make our life more difficult and try to do this in mammalian cells. Obviously, great strategy. But forego this portion of trying to understand too much. So rather than trying to have a perfect computational model, let's try to come up with a black box model, but that is not too simple either, because the black box models that I've actually tried to use in the past didn't work either. Okay, so really try to respect a little bit more of what the black box model should be. How much information do we really need to have in order to hopefully be able to do these kinds of predictions? And hopefully these black box models will also capture the cellular context, the interaction with the cellular context. How does that cellular context then affect the behavior of each one of these parts? So characterize, so this is a part that we characterize. So we have a simple repressor. So we have an input, we activate a repressor, and we see what happens. In the output, we have a constitutive fluorescent protein, input, and then output fluorescent markers. And so these are the transfer curves that we generate. And notice that this is not your typical transfer curve. So we have a relationship between input and output, but we're also noticing the fact that I mentioned large heterogeneity in the way our circuits function in mammalian cells. And so this represents the constitutive fluorescent protein expression with transient transfection. You see huge variations. So there's no way we're going to be able to really predict what happens unless we try to understand a little bit more about what happens to the circuit in the individual cells. So what we do is we bin it so that we take each subpopulation based on the expression level of the constitutive fluorescent protein, and now that approximates some kind of a circuit copy number. And so each one of these lines now represents the transfer curve, the input-output relationship for a given circuit copy number. Okay, so this look is at the tau repressor 14, that's one of the ones that we made, and the 21. So we made a library of 30 of those, and we characterized a few of those. So now we have a relationship that not only tells us input-output, but also looks at the effect of the circuit copy number on this. And so then we said, okay, let's put these two repressors together, and this is what the circuit looks like. It's a cascade, transcriptional cascade, and ask, so let's build these transfer curves and then create models and then see what happens. So I like to always show this, results that don't work, because we get a lot of this. I want to share this with you. So here's one that we created an OD model, and you see the predictions of the cascade versus the experimental result. So even though we're getting kind of the general fact that there's a correlation that goes up as the input goes up, the output goes up, not quite where we need to be in terms of engineering. So the ODE model that we created, even though we had much more information, was not quite sufficient. And so we said, again, let's go back to this black box model, but have a better one where we have, kind of respect the black box a little bit more. So we look at production rates, we look at dilution and decay, so build kind of a more dynamic model of what the black box is, as opposed to just trying to map input-output steady-state behaviors. So really try to understand what's the incremental behavior of each one of these devices. And indeed, once we put together, and there's a lot of work that went into this, this, I'm showing you this quickly here, but there's 15 different things that we had to actually go through to get the details right. But eventually, we are getting very good predictions. So you see here the predictions, that's the lines here versus the actual experimental results. So it's very nice correlations. So now the mean prediction error is only 1.2x for over 300-fold variability in the actual experimental results that we're seeing. So this is now getting to the point where predictions appear to start making sense and maybe actually useful for creating larger and larger circuits. So we did, as I mentioned, need to create much more detailed black box models, but that seemed to have helped out a lot. And one of the things that we eventually want to be able to do, and then go back to the more detailed biochemical models and see whether these kinds of analyses can actually help us then develop a better biochemical model of the processes that are taking place. Predictions would be nice, for example, in some of the applications. So this is an application, this is one slide that I have about this work that was done by Zhenji, and this is in collaboration with Kobe Benenson at ETH. And so here what we did, and Jen is actually here today, so here what we did is we identified that one of the major problems with cancer therapy right now is specificity. Right? So we can get therapeutic agents that can kill all the cancer cells, but oftentimes they have huge side effects on the patients. Right? And so if we can increase the specificity, this would be obviously clearly very important to be able to increase the dosages and be able to deliver the therapeutic agents to all the cells and have it just act only in the right cells and not have any kind of collateral damage. And so what synthetic biology can add to the mix is this notion of computation. So in particular, we would argue that you can't look at a single biomarker. There is no single biomarker for cancer, right? For just about all, most if not all cancer types. Especially when you're talking with heterogeneous populations and you get mutations and so on. But if you start, kind of respecting cancer in terms of its system-level behavior heterogeneity, you can develop cancer profiles. When you look at, for example, we would argue, whether it's 6 biomarkers, whether it's 10 biomarkers, or so on, something manageable would then be able to give you an incredibly precise determination of whether that individual cancer cell, whether that individual cell is cancerous or not. And so 6 biomarkers for HeLa cells is what we came up with. So three of them have to be high in some combination, 3 of them have to be low, and if the answer is yes, that's a HeLa cell. And this is the circuit. I'm not going to go into the details of the circuit. You can read about it here. But it looks at microRNA levels. These are the high microRNA sensors, this, and these are the low microRNA sensors. And we get a high output only in the situation where it matches the profile. And we tested this in terms of red fluorescence expression in HeLa cells actually against other cancer cell types, and it seems to give us high specificity. Afterwards, we also created a single plasmid version of this. This was done with a 6 plasmid co-transfection. We created a single plasmid version of this, and we transfected that, and we saw that even at the single cell population against the worst behaving cells, which were the HEK cells, we get essentially complete separation at the single cell level between the HeLa cells and the worst case behaving cells, the HEK cells. So we're quite excited about the technology, and even small, quote unquote, engineering things allow us to get the specificity that we want. We also tied in a killer protein to this, and we put this again against the worst behaving cell, the HEK cells, in a mixed population, and it was really able to selectively kill the HeLa cells and really not have any serious impact on the HEK cells. So one of the things that we're working on a variety of extensions on this. One of them is safety. And so let's see here. So this notion of delivering DNA, and many of us are interested in being able to deliver DNA therapeutically, but perhaps RNA for a variety of applications actually can serve as a safer vehicle to introducing genetic material into the cell. And so this is one of the things, we're still working with DNA obviously, but we really are excited about the possibilities of being able to encode complex circuitry on just RNA delivery. So the only thing you deliver is RNA. All the regulatory interactions happen at the RNA or above level. And then hopefully this notion of integration, chromosomal integration, is not really a factor anymore. The RNA stays in there for some amount of time, does a therapeutic purpose, and then eventually it will go away. And so for that, this is Leela in my group and various other people, and Tasuku as well, are working with RNA-binding proteins. So we deliver RNA, and then these RNA-binding proteins interact with aptamers. So they bind, and then they translationally repress production of other proteins. So here you see, and this is a circuit that Leela was involved in the previous cancer study. This is a circuit now that's basically the same as the HeLa classifier that I showed you with DNA. This is now an implementation with RNA only, using RNA-binding proteins here. And you see here that we have very nice on-off gain in their behavior. So they work quite well in terms of being able to then construct larger and larger circuits out of them. And so this actually works. I show you a result from a portion of this. And we said, why not build other kinds of circuits, because they're always fun. And so I'll show you quickly results with a cascade and some beginning efforts of a memory switch that's just based on RNA and above. And so I guess the computer really wants me to be next to it for some reason. So here's the microRNA sensor that we built. So you see here on-off ratios with microRNA sensor that works quite well by interacting with an RNA-binding proteins. And then, as I mentioned, Leela has also experimented with a full circuit. Seems to be working. Here's a cascade. So now we're hooking up together multiple RNA-binding proteins that are able to sense the microRNA over here. And you see here, basically, as we build the elements in the circuit, so this is just this element right here. We see high expression. We had a repressor low. We had another repressor that goes high. We had the microRNA sensing, and that goes low again. So we have, and we're getting very nice on-off behaviors just with the introduction of RNA. And also we have some preliminary results suggesting that when we cross-repress these RNA-binding proteins, we can set the switch one way, or we can set the switch the other way. So it does seem, the RNA-binding proteins do seem like a viable choice for being able to create interesting circuitry out of them. Okay, and the last thing I want to very quickly mention is work that Patrick's, I'll talk about a couple of things that he's doing with stem cell differentiation in this collaboration with the Church Lab and Zheng Lab over here. So this is one where we'd like to be able to program tissues by design. So be able to create circuits, putting them inside iPS cells. It could be patient-specific cells, and then be able to regulate their differentiation into any cell type that we want. And so Patrick, what he did here, and this also worked with Yingqing in my lab, is he created a simple circuit where we have Dox induction of a couple of neuronal differentiation factors, NGN1 and NGN2, and just showing that you can take these iPS cells and we create very nice results where we have relatively pure populations. I think based on some estimations, 85% or more have differentiated into neurons within 4 days. And from what Patrick tells me, this is significantly better than what's out there right now, where other competing mechanisms take more than 15 days and give you 25, 30% efficiencies. And they seem to be immature neurons that actually have some ability to activate. And now Patrick with Volker, the Church Lab, have also done RNA-seq to try to understand what is the process that they're going through. Okay, so there's some really interesting information about, from the RNA-seq in terms of what things get upregulated, what things get downregulated. And Patrick has a poster about this, just in case you're curious about some of these lines over here. And the last thing I want to briefly mention is not only, not only do we want to be able to genetically program these pure populations. So one of the things that we've been interested in very recently is this notion of genetically engineered multi-cell populations. And really, up until recently, we focused more on getting these pure populations to differentiate into particular cell types without fully appreciating that a single cell type has a hard time becoming the final terminal cell type of choice. And so the question now we're trying to explore is, can we genetically engineer multiple populations of cells so that somehow the interactions between these multiple cell types is much more biological, right? They much more depend on one another to create the tissues of interest. So Patrick did some work with, and again, this is on his poster, taking human iPS cells and then creating multiple versions of these, which now exist as mixed populations, induce them, and so then they kind of go in lockstep in terms of the differentiation. So go into the endoderm, and that results in further differentiation. And we've seen some very interesting results with respect to structures that may look like some components of liver. So they actually have some liver properties where we now have these hybrid engineered tissues that have both hepatocyte markers associated with them. And again, Patrick has done quite extensive study on this, as well as mesoderm. So multiple cell types that have been engineered to work together to create the final tissue of interest. And so with that, just very quickly summarize. So we're working from the foundational elements of, can we build big things? What are the particular regulatory modalities that we think would be fun and perhaps very beneficial for particular applications, for example, RNA binding proteins? How do we optimize the workflow so that we take high-level ideas and then maybe sometimes can actually predict what would happen? And then come up with a few application areas where we think synthetic biology can make really a fundamental difference. And so with that, thank many people in my lab. I mentioned most of these during the talk. Funding agencies, DARPA, NSF, and so on. I have Princeton in there. I could remove that, but they did help me. And I want to thank you as well. Thanks.
Excuse me, my name is Chris Smith. I'm from Johns Hopkins University. I had a question specifically about the area when you talked about looking at profiles for the different types of cancer. So I know that there can be a lot of issues with variability within a population and outside the population. Yeah. When you were trying to develop it, did you have difficulties with the variability within the population? And also, when you were developing profiles, did you look at different cell lines of the same cancer type?
Yeah, no, that's absolutely a great question. So for the initial implementation that I showed you, basically looked at what was known from the microRNA atlas about mean levels of microRNA activity, and that was just a start. And so recently, we've become very interested in understanding single-cell statistics. So everything that we gather is now based on single-cell statistics. So try to understand what are the distributions, and we have all that data for the various cell lines that we're working with. But also try to understand single-cell correlations among multiple microRNAs. So one of the things that you mentioned, which is, I think, incredibly important, is within a homogeneous population of cells, you may have some dominant populations, but then you're going to have some subpopulations. And so we have projects right now to really be able to quantify what are these subpopulations? What is that 1% subpopulation that, if you develop a cancer therapy and you got 99%, that's really not sufficient. And so the modifications that we're working on now to these cancer classifiers will take into account these very small subpopulations and develop circuitry that can identify them and kill those as well. Yeah.
Thank you.
Hi. Ann Plant at NIST. That was really a very fun thing to listen to. And I happen to like distributions and broad distributions. So okay. I'm curious if you are taking you know, if you're looking critically at why some of your circuits give you narrow distributions and some give you broader distributions, and if you're coming up with any general rules about why that occurs.
Yeah, I mean, it's a great question. I think in general for, you know, whether you're doing bacteria, whether you're doing mammalian cells, whether they have, you know, different reasons. So I'm not, you know, sure whether we've identified different reasons, for example, from bacteria to mammalian cells, the different chassis. I mean, we certainly have ideas about specific network motifs and how those specific network motifs may influence the distributions in the populations. And so we've done some studies, you know, more in bacteria to look at, people talk often about negative feedback as a way to attenuate noise. Not that simple. So it really depends on the particular negative feedback. What are the dynamics, but also what is the strength of the negative feedback? So the same network motif with feedback, negative feedback, sometimes gives you lower noise and sometimes it gives you much higher noise.
Right.
So I mean, it's an incredibly rich question. I mean, with respect to some of the things I showed you, you know, the transient transfections obviously, you know, give you large heterogeneity. Yeah, Kobe Benenson, for example, had a paper showing how you can create microRNA motifs that even for transient transfections can actually reduce it. One thing that I like, you know, with Pam mentioned also with respect to building things that operate more on the RNA level, right? So, you know, if you have large populations of RNA molecules, will that tend to reduce noise in the system?
Yeah, I guess that's part of the system. Is it, you know, a function of the size of the system, the numbers of molecules involved, or the motif of regulation? Right. Yeah.
And the other thing that I care about is also, you know, sometimes we're kind of stuck with noise. And so we're, you know, we often as engineers think about let's attenuate noise and really try to get rid of it. But something else that I care about is how can we actually benefit from noise.
That's exactly right, because it is the essence of biology after all.
Yeah, and so we published a paper about a year ago about some theoretical pie-in-the-sky system that for artificial tissue homeostasis. And we ultimately want to use it, for example, for beta cells and other cell types, where the system actually behaves better when you have noise. And so we actually we have specific mechanisms in there to induce noise in the system, so you get better overall multicellular performance.
Okay, last question.
Hi, I'm Faisal Reza from Yale University. That was a great talk, Ron. I'm actually following up on the question about the cancer cell circuitry. We found in our work when we try to engineer the genome that if we marry the internal engineering with sort of exogenous cues, that we can get better output and more sensitive output. For example, with marrying hypoxia to internal response. What are your thoughts on that?
No, I completely agree about that. You know, we want to, you know, one thing we often do is we think about the circuits as, you know, these isolated entities, and especially in bacteria, you know, and that's all nice, but it doesn't really work. So in terms of understanding how the cellular context affects it, and also the environmental context, you know, an active area of research. I have a collaboration with Domitilla Del Vecchio on quantifying the effect of the cellular context. How can we use that to actually engineer more robust, you know, networks? So I think it's really critical for us as a community to have computational, you know, understanding of the cellular context and environmental cues that can actually help. And I'm going to pull in a plug for Gregory Bott, who's right behind you, who did some really nice study of creating computational models of synthetic gene networks and interaction from the outside, for example, light induction, to be able to have the population really follow much more closely a desired gene expression pattern. So, and he can give you a lot more information about that exciting work.
Excellent, thank you.
Thanks, Ron. Okay.